I think we're going to get started here. I realize it's been a long day, but exciting, and I know we're the last group to keep you from the reception, but it's a good group, and we're excited to get started. So this is session 5, which focuses on the patient, clinician, and workflow considerations as it relates to incorporating integrated diagnostics and precision oncology care. My name is Anna Naik. I'm a professor in the Guinée Chair of Geriatrics at the University of Texas Health Science Center, Houston. And my co-moderator here is Jason Slagle. He is an associate research professor at Vanderbilt University Medical Center. Jason and I will introduce each of the speakers one at a time, and we're going to start with Dr. Saurabh Shah, who is the Chief of Computational Oncology and the Nichols and Bondi Chair at Memorial Sloan Kettering Cancer Center.
Thank you very much. Thank you for the organization of such a great meeting and for inviting me. My talk I think is going to be a little outlier in this session, but I think it relates very closely to a lot of the topics that we've heard throughout the day. So my disclosures. Great. So I wanted to frame this by discussing the frontier areas of computational oncology research. And I think I want to place this talk very much in the research domain because I think we don't have clinical implementation until we have new insights that derive from research. And so here I'm listing a few areas that are exciting and we've heard a lot about today, insights from real world data, which is data that's procured in the provision of care from a patient. Multimodal data integration, which will show you some examples of that. Large-scale cohort derivation and comparison. Can we discover cohorts that we can actually then analyze and juxtapose against each other to learn something about what's different between these different groups of patients? And now ultimately, of course, patient stratification and predictive models. Once we build these models, when I see my new patient, how can I get a sense of how they might respond to a given therapeutic intervention? So we've done a lot of work in this area. Here I'm listing a few papers. The top one is a review paper that we wrote that really starts to articulate how we can bring these different modes of data together to build an integrated predictive model for response to a given therapy. And then the two papers underneath are really examples of this, one of which I'm going to show you a little bit about today. At Memorial Sloan Kettering, we've been very lucky to have had a long-standing investment in building platforms, and Nikki Schultz has really led the charge on this in building, for example, the CBio portal, which is an integrated database for cancer mutation data. Charles talked about it, but there are these other efforts that have been implemented over the last few years that are now being put together under a single initiative called the Cancer Data Science Initiative at MSK. And I think this is really going to propel research forward since we can bring together in a unified framework these disparate multimodal data sets from patients. So that's the real world data. On the right-hand side now are just incredible developments in experimental data from which we can apply new technologies to understand key principles in cancer biology, cancer evolution and drug resistance. We know that a tumor is not a static entity. It is a collection of divergent cellular populations that when selective pressure is applied will have different responses. So in order to understand how a tumor is going to progress, we really need to view it through the lens of cancer evolution. Of course, cancers then evolve in the context of their tumor microenvironments, and they acquire different cellular phenotypes. It's a dynamic process within one patient, even before treatment in disease natural history, and then after treatment with the selective pressure. And this invokes the idea that tumor cells interact with immune cells, and this has major implications, of course, for designing cancer vaccines, new immunotherapies, et cetera. And ultimately, this, in order to get a very clear picture of this, require multi-omic integration. So here are some of the experimental data types on the bottom right that we're going to have to grapple with. These are research tools at the moment. for discovery. But the pace of discovery is ferocious in this domain, and I think it's very exciting. So I think what I wanted to do is stimulate thought about how do we incorporate new innovative measurement technologies that are really research tools, but the results of those research should be thought about as a pipeline for integrated diagnostics going forward. Okay, so that's all the vision part. I'll walk you through this study that's been referenced a few times today already. This was one of our first projects that, when I arrived at MSK, decided to put my hat into the field of real-world data integration, multimodal integration. I basically set out a request for applications to all the disease management teams, and the lung tumor group responded and said, we have this problem. We have this cohort of patients that we're following. It's about 250 patients have been treated with immune checkpoint blockade. But we're just very poor at predicting who's going to respond and who's not going to respond. And so could we learn something from layering on all the different diagnostic measurements? We have genomics, we have PD-L1, immunohistochemistry, and we have radiology. So we set out to create an automated system to extract features from each of these different modalities and then integrate them with a machine learning model that could pull all the different features and the different modes together and adaptively weight them to provide the optimal prediction in terms of whether a patient will respond or not. So here's an example, though, of the labor that's required to do this. So in order to do this properly, we had to scan all the immunohistochemistry slides, and this was done in collaboration with Jennifer Sauter, who's a thoracic pathologist. And then she labeled all the tumor regions in these digitized images. And from there, we then started to compute the topological features of the staining properties within each of the tumor regions that she labeled. So this was about, I would say, a 1.5-year process working with Jennifer and iteratively doing AI and feature extraction from that process. So this is a specialist pathologist physician that was interested in doing this work with us, and that's what made it possible. I'm just telling you very frankly that we wouldn't have been able to do that without her. So the result of this, and there's an analogous process that happened in radiology, but the result of this, when we finished, was the following thing. So these are quartile stratified outcome curves that are really trying to divide the population into risk groups. On the left is the clinical PD-L1 scoring, and you can see that the risk groups are not well separated. Next over is tumor mutation burden, another on-label indication for immune checkpoint blockade. It's a little bit better, but not fantastic. And then on the right is the integrated model that brings together genomics, histology, and radiology, these computationally derived features from these raw data files from the diagnostic tools. So that's exciting. That's a proof of principle. And the hypothesis is validated here, but it's not too surprising either. The idea is that we're measuring a complex system. You do this through different tools. Then when you layer on that information, the picture should become cleaner. That's just the basic principle of statistics. You have a complex system, you take multi-scale measurements, you pull them together, you should get a better prediction. And that's what we did. So then on the research tool side, this is now, you remember that original slide I showed, now we're firmly in the right-hand side. So we wanted to learn a little bit more about how ovarian cancers actually have, we've known for a long time that there's a wide range in immune response in the natural history of disease in ovarian cancers, pre-treatment. We also know that ovarian cancer can be stratified according to the DNA damage response profiles that are present in their genomes. So we can read out the genomes of these tumors and then actually ascertain whether a patient is homologous recombination deficient, has a tumor, or is a homologous recombination proficient tumor. This has all kinds of implications for stratification onto PARP inhibitors and other therapeutic modalities. And in particular, what's been surprising about this disease is that they've been woefully disappointing in single agent immunotherapy. So we wanted to try to understand what's the interaction between the genomic DNA damage footprints and the immune response in this disease. And so, again, I would just stress that in order to do this work, we collaborated with the whole disease management team from surgery. We collected samples. We undertook a workflow where we generated single-cell suspensions from these surgical specimens, disaggregated them into, and then flow sorted, and then in a single-day workflow, executed single-cell RNA sequencing on this to profile the phenotypes of immune cells. So, and in conjunction with that, we did whole genome sequencing and then multiplexed immunofluorescence. We bring the data together and we get a picture that really tells us something, that the underlying mutational process of this disease has a major bearing on the immune phenotypes that are present. So without going into more detail, the paper's published, you can read it. But the first point is the one that I wanted to say, that multi-omic views are really central to answer questions about disease biology. So how do we do this? And I'm going to be wrapping up very soon. Is to do this at scale needs significant data engineering efforts. And we need to be able to pull data from source into a common data lake, have tools that we can easily query the data, and then actually execute machine learning research on that. And so the idea is that there are no copies of the data. We just have a central source. We give access to researchers and physicians alike, and hopefully have large-scale compute that can then develop these types of models and inferences. So just to wrap up quickly, I've just put up a few things that I think are important. Multimodal data integration is a very powerful approach for both real-world and experimental data interpretation. It's a team sport, and it really requires multidisciplinary collaboration that is at the interface of clinical expertise and computational rigor. And the path to integrative diagnostics, I think, will be informed by large retrospective multimodal studies, but this field's very early in its development. We're only understanding now how does a pathology image relate to a radiology scan, for example. So I think with that, I should wrap up. Reproducibility is required for these models, but it's hard. And so we need to be able to validate models across different institutions. And finally, talent is a bottleneck, and we need to fix that. So with that, I'll stop.